So once we have RNA polymerase transcribing the gene and making a messenger RNA copy, at some point it comes to the end of the gene. So how does it know where to stop? At the end of the gene is another sequence. In the same way that we had a special sequence to show RNA polymerase where to start, there is a special sequence showing to RNA polymerase where to stop. And essentially it is an inverted repeat sequence. Here is an example of an inverted repeat sequence. Here the A's match up with the T's, the C's match up with the G's, and so on. And they fold up. They can fold up into something called a hairpin loop. That means here's a single strand, a piece of RNA that folds up into a loop, and there's base pairing in this stem, or this hairpin loop around here. Once this occurs, then RNA polymerase will fall off if it is stopped on a sequence that is rich in A's and T's. And why is that? What is special about the bonding between A and T compared to between C and G? If it's not on an AT-rich region, a protein called Rho will be nearby, and that will physically knock off the RNA polymerase, and therefore stopping transcription in that manner. So we have Rho-independent termination with AT-rich sequence, and Rho-dependent termination if it's not an AT-rich sequence. This is a diagram of what a stem loop looks like. As you can see here, we have a sequence on one side of the single-stranded RNA, loops around, comes back on the other side, and bonds in the middle. So this is a stem loop, also known as a hairpin loop. Here is the bigger picture. This is the DNA double helix through here. This purple blob is the RNA polymerase, or RNAP in here. And this is the termination sequence just about here, well, and down here. So this is the hairpin loop, and this is the ending sequence here. So this hairpin loop will form just after this messenger RNA has been made by the RNA polymerase. What that then does is that this hairpin loop basically folds over and stops RNA polymerase from moving. Once you've made your messenger RNA message, we're going to modify it in a number of ways. And there are three things that we're going to do to the messenger RNA. RNA molecules contain introns and exons if they're messenger RNAs. And the way you can remember which is introns are intervening, and we want to splice them out, so we want to remove the introns. And the exons, we want to keep them because they are expressed. So introns are intervening, we want to remove them. Exons are expressed, and we want to keep them. There are three things that we're going to do to your messenger RNA. We're going to cap it at the 5' end. We're going to remove these introns in the middle, and we're going to add a poly-A tail at the 3' end. Let's do the capping and tailing first. When we cap a messenger RNA, we're going to add a base to the 5' end. So here's the messenger RNA, 5' end on the left, 3' end on the right, and we're going to add a base to the 5' end. It's a single base, and it happens to be a guanine molecule. So we're going to add a G to this 5' end. The slight difference is it is not a normal G. It is a 7-methyl guanine, so it's been slightly modified by adding a methyl group, and we're going to add it backwards. Instead of being a 5' to 3' addition, it's going to be a 5' to 5' addition. So the cap is a 5' 7-methyl guanine molecule. Here's the diagram for this. This two green lines are the double helix. This red line is the messenger RNA molecule, and if you look over here, here is the 7-methyl guanine molecule that is added to the 5' end of the message. Once we've capped the molecule, we're going to add a poly-A tail that's down here on the bottom right, and if you notice here, the RNA polymerase, this is RNA polymerase, the RNA polymerase continues to transcribe and continues to make messenger RNA. At some point during that messenger RNA, the molecule is cleaved. Even though RNA polymerase continues to go on to make some messenger RNA that was no longer functional, that message is cleaved. That cleavage signal is given by another special sequence. That sequence is AAU, AAA. Just downstream, just 3' to this cleavage signal is the cleavage site, and that is where the messenger RNA is cut. It's only about 10 bases or so just downstream, so this is about 10 bases down here. And an enzyme called polyadenylase adds a whole bunch of A molecules. So this is polyadenylase. And it's an enzyme that adds lots and lots and lots and lots of A's. And you can see here, and there's roughly about 200 adenine molecules. And this gives us our poly-A tail. So let's get back to this splicing, this removing of the introns. Here is a messenger RNA molecule, and you can see it's already been capped and it's been tailed, and we're going to get rid of the introns, and we're going to keep these darker colored exons here. And here is the mature messenger RNA transcript, and you can see all the exons are joined together and the introns have been spliced out. So we call this removing of introns splicing right here. And how do we know where to remove introns? Well, luckily, all introns begin with the sequence GU, guanine uracil, and all introns end with the sequence AG, adenine guanine. And so the enzymes that recognize introns are looking for a number of sequences, one of which are the GU and AG consensus sequences. The enzymes that are cutting out introns are called SNRPs. Stands for small nuclear ribonucleoproteins. Don't worry about it. And the interesting thing about SNRPs is that they are ribozymes, and ribozymes are defined here. Ribozymes are molecules of RNA that have enzymic activity. So here is an overall diagram of an intron with its surrounding exons. This area to the left is the left-hand exon. The area red to the right is the right-hand exon, and this lighter area in the middle is an intron. And if you look, here is the GU sequence, here is the AG sequence, a whole bunch of other consensus sequences in the middle, but the GU, AG are what we're interested in. And this intron sequence here, and these are the exons, both sides. And as you can see, the intron folds up, is joined to itself, and then the two exons are joined together to make a continuous expression piece here. So this intron is now folded up and then destroyed. So here is a bigger picture here. Here is the gene. This is DNA across the top. Here is the promoter region on the left hand side. Here is the expressed region. Here is the first exon. Here's an intron with the GU and AG consensus splice sequences. Here's the second exon, second intron, GU, AG, and so on and so on. And here is the end sequence with the poly-A addition site, the AAU, AAA. And transcription termination occurs just after that. We can summarize all of this into one diagram, which you would be well behooved to learn. And this is all across here. This is a gene. This is basically DNA across here, and it shows the promoter, the first exon, an intron, second exon, translation termination sites, polyadenylation sites, and so on. You should be able to draw an entire gene and all the components of a gene and explain what those components do. So if we have more than one intron, that gives us the possibility to remove an intron in one version of the messenger RNA, but leave it in another version of the messenger RNA. So our splicing could be variable between different messenger RNA molecules of the same gene. And this is an example. This is the rat alpha tropomyosin messenger RNA gene. And this raw messenger RNA across the top here, we can see that there are a variety of different types of muscles that are made, muscle proteins made from this one messenger RNA. These are the exons 1A, 2A, 2B, 1B, 3, 4, etc. And as you can see in the first version here, exon 2A has been removed. So we don't have exon 2A. But in the smooth muscle version, we do have exon 2A, but not exon 2B. And in the brain versions of this protein, exons 1A, 2A, and 2B are all missing, and we start with 1B. And you go on and so forth. We have all these different types of muscle proteins that are being made from this one particular messenger RNA. And this is called alternative splicing. And this is a really important concept to get, because this can explain how humans can have hundreds and hundreds of thousands of different proteins when you only have 20,000 genes. Multiple proteins from one gene.